Welcome to iBOL's new webinar series, BioScan, Illuminating Biodiversity. The title is a bit longer, so it's called 9 Malaise Traps in 3 Costa Rican forest hectares catch 40,000 mostly undescribed species of insects among 1,521,762 plus specimens in 7 years. Now what? Dan and Winnie have long been tremendous supporters of DNA barcoding and iBOL. They are tropical ecologists and biodiversity conservationists and co-founders of the Area de Conservación Guanacaste, short ACG, a network of protected areas and a World Heritage Site in Guanacaste province in Costa Rica. Jan is also a de Mora professor of conservation biology at the University of Pennsylvania. Winnie has, among other things, worked as a researcher and technical advisor to the ACG. Dan and Winnie have implemented various biodiversity projects and programs and have a combined 109 plus years of field experience and have written more than 600 scientific papers and books, all focused on the interaction of tropical animals and plants, and for the past 37 years on their permanent in situ conservation project. A full bio doing justice to both their lives would probably fill a webinar on its own, which is why I will stop talking now and leave the floor to them. Dan, please take it away.
All right, good morning, everybody. This is a very complicated talk. It deals with many different things. It starts at the very high level with BioScan, which is, of course, as I think many of you realize, a global effort to develop a barcode library over the whole globe. And that obviously is going to be a thin layer over the whole globe. What we're talking about today is a deep penetration through that layer for one country. It's ended up, the activity has ended up with the name of BioAlfa over here on the left. And I'm going to get my pointer on going over here on the left, BioAlfa. And that will become clearer as to what that is as we go along. The very specific, right in the mud level question at the very bottom is, as Dirk has just said, we have ended up with 40,000 undescribed species amongst 1.5 million specimens over 7 years from one place, one 3-hectare place in Costa Rica with from 9 Malaise traps. That's just a tiny, tiny sample of what we're dealing with. So the question becomes, what do you do with a piece of dirt of this nature? Just the chunk specific stuff. Well, first off, you begin to recognize that it's not just an ivory tower exercise. It's part of facilitating the whole country of Costa Rica into becoming bioliterate. Now, the title for this area of activity is BioAlfa, which is obviously a derivative from the Spanish word for bioliterate. This slide is meant to emphasize that the government is part of this, the entire government of Costa Rica, the Ministry of Environment within that government, the Park System Service within that Ministry of Environment, and then over here on the right-hand side, the full regulations of the government legal structure behind it, then an NGO, GDFCF, which is dedicated to the development of the specific site where this is occurring, the Area de Conservación Guanacaste, or ACG, as you'll hear me say, and then finally the title. And this insect is chosen because everybody in Latin America knows this insect and its story. This was not believed by people in the United States. It's not a toucan, it's not a monkey. So one of them in California went out and asked somebody working and basically in the fields in California and asked him, do you know what this is? And he said, oh, yes. Where are you from? Guatemala. So there we go. So where is Costa Rica? It's right here on the globe. And by quite fortunate turn, here is the globe itself. But this is in the Toronto Zoo with a Canadian flag, who's been a major supporter for this activity from the very beginning. So biodevelopment is really what we're talking about of the wild biodiversity instead of turning it into standard agriculture. Now, what is the biodiversity of Costa Rica? Well, here's Costa Rica, a tiny little piece of terrain. And here it is on the United States for scale. It's nothing. It's just a teeny piece, okay? There are counties in the United States which are bigger than Costa Rica. But if you measure it by its biodiversity, there's 4% of the world in Costa Rica, and there's 4% of the world in North America. Down at the country level, all the white areas here are national parks or the equivalent, and the yellow is also a national park and its equivalent. But this is the ACG, the Área de Conservación Guanacaste. And that's where we're focused, is on that piece right there. 60% of Costa Rica's biodiversity lives in this 2% of the country. I'm not saying it can survive in the long term there, but that's where it is at the present time. So let's look closely at that. Here it is on the ground, a topo map, basically, of the ACG and Pacific Ocean here. And we're focused on the terrestrial side today. Dry forest here, cloud forest at the tops of the volcanoes, and rainforest over on the Caribbean side. So that's the basic structure. And we go and take a Malaise trap, which you all know, I'm pretty certain, and we put that at each of these points, there, there, and there, for 1 year each. What do we get? What we get is 19,000 species from 3 traps in 3 years. But each one of those is a slightly different year, but that's all there were there. So now in this are their numbers. So they're 5288. That 5288 is the number of bins, and we'll get to what bins are in a few seconds, in the dry forest trap. And we estimate this to be about the number of species from that trap. So this is the collective. Now the point about this is that these species, 19,000 species, only have 31 bins in common. We're talking about 0.2% in common between those 3 traps, which are only a few kilometers apart. So let's look at what are bins, because they're going to be used all the way through this. This is a standard NJ tree produced by iBOL at the Center for Biodiversity Genomics and the University of Guelph in Canada. And this particular tree has in it, and I'm realizing here I've got a blockage, I can't read my own stuff. So I have to move this and see if I can move that down. I can't. Okay, well, up here at the top somewhere, it tells you how many species there are in this tree, and you can see also how many pages there are on this tree. So this is an enormous number of specimens of one fly family who are parasites called tachinidae. And here's an actual tachinid laying an egg on this caterpillar. So when we talk about reared tachinidae, we're talking about those who we have reared from caterpillars in the ACG. So here's the NJ tree over here, and each one of these is one record. And as you can see, there's a cluster of them right here, which has got a name on it. And over here is a code, which is the name for that bin. Okay, so think of that, if you like, as a species name, which is here, which is applied by a proper taxonomist. And over here is the bin record from BOL. It tells you that they're all the same thing as well. Now, notice over here, it says Huhia Malaise 833. That is a code, a nickname, if you like, put on it by basically this is an arbitrary number, but the point to put on it by us using the clustering here to guess that this is in fact one name. So before we had bins, this would have been cluster would have been called one thing. And now we have bins and cells, which are based on about a 2% difference amongst these different species. And so you end up with a little cluster here of 3 individuals. And that's the same thing as a classical taxonomist museum taking those 3 and putting them in one unit tray, which later gets a name because they look like each other. Well, here they look like each other. They have the ecologies like each other, and they have a DNA barcode like each other. And then finally, we have one down here that says, and these 3 were caught in a Malaise trap. These here are reared because they've got my code in there like that. That tells you they're reared. So these are Malaise traps, and these are reared. And as you look down this line, there's a lot of reared ones, but down here are a couple more caught in a Malaise trap. And the way we do basic identification is to take all the stuff from the Malaise traps and throw them into the same tree with all the reared ones, and then see how they fall together. Oh, somehow we're not moving forward. Something seems to have happened. I don't know what to do because it's now frozen.
What if you did this?
No, I have to get rid of this thing here. No.
No, now you're fine.
Okay. We think I'm unmuted. All right. Why is there a big difference between the number of species and the number of bins? This number here is always much larger than this number here. That's because a number of bins have multiple species in them. That's just like the old time, today still, of course, taxonomists looking at things and saying, oh, these all look the same to me. So it puts them all in the same unit tray. We then barcode them and discover that they have got differences in their barcodes, and they've got differences in their biologies. And just over here on the right, for example, this box up here on the right, top right over here, has got 11 species in it that we're hiding under one name. This box right here has got 3 species in it, we're hiding under one name, and this has got 4 species in it who are hiding under one name. For some reason, I am not being able to advance my PowerPoint. So I'll do it with these arrows down here at the bottom. Now let's go back to our view of the whole ACG, and these are where the Malaise traps were, and ask about something that could be of very much direct interest to the people on the street, to ordinary people, not the academic viewpoint of how we classify these organisms, how we identify these organisms, but what good is it? Where does the use come? Well, you see this yellow star over here. That yellow star is at a point where at the base of this volcano, where the value to of the society as a whole is that underneath that star is an enormous pot of boiling water. And basically what they want to do is stick a straw into that to pull the boiling water out and use it to run electric generators. So this is where that site is. And notice that this is our boundary, the boundary of the World Heritage Site, the conserved area. And this is owned by the state, this forest here, and this is owned by the state. But this is outside and up for grabs and up for use, all right? Well, they want to drill right here. So we, listening to this plan, said to them, what, can we work together on this? Your industry, we're conserved. After a lot of thinking, they decided, okay, but we don't go public about it. All right. So this is where BioAlfa comes in, because BioAlfa's goal is to not only make the DNA barcode library or facilitate it to happen in Costa Rica, but also to put it to work. We have lots and lots of insects, so let's put them to work. So they give us the blueprint for what's going to happen. The road through here, through the forest, the chunk of a couple of hectares cut out of the forest here for the geothermal project. And so we look at that and say, what if NSF had given us $5 million to do a research study of what happens in a tropical, very complex forest if you drive a road through it. If you put a hole in it of 3 hectares, what's the reaction? If you try to ask a mountain lion or a monkey or a toucan, what's the reaction? There's no way in hell that you can get that. But you can use community structure of insects as reflected by trapping to measure the impact or the response by that system. So you get to start. You're right there when the bulldozers go in and do the first horrible looking job on that very nice rainforest. And there's your Malaise trap right there. At the same time, the bulldozer has been doing all the clearing and all that. A very short time later, they prepared the site. Your trap is still right there. Now they're drilling and doing all their real polishing up work. Trap is still right there. And 7 years later, it's now an operating site. And your trap has been right there all the time. Another one 50 meters into the forest, another one 150 meters into the forest, and 3 lines like that around this place. One here, one over here on the right, and one on the road going to this place. So that's where we are. So that's our focal trap, and we'll just take that one right there. And here it is 7 years later. Now it's been changed once during those 7 years. So this is not the original trap, but rather one that came on after 3.5 years. There are 9 of these traps around this platform. This one in 7 years has got 25,000 species, 25,000 species of insects. This is what they look like. So this is just that one trap over 7 years. There's the rainfall data provided by the electric company. There's the temperature data provided by the electric company. And this is a summation of all 9 traps. So this one here finally ends up at 40,000 species from that one place out of this number of specimens. Okay. All done by barcoding. Built on the taxonomic structure that's already received by BOLD from the identified things put in by, shall we say, standard taxonomy. Indispensable part of this operation. But it's done by layering the barcode data on top of that taxonomic structure. So what does the geothermal project care about that? Here's where that trap is. There's another edge right here. And just 50 meters into the forest, there's no recognition of the existence of this platform. At 150 meters, there's no recognition, no perturbation of the insects. But of course, here there is a big change on the edge. Here's what those 3 traps way inside the forest look like over the year, provided by barcoding all the specimens from those traps. These are the number of species caught each week, okay, against the temperature system for the system. And here's the rainfall down here, and here's where the drilling takes place. So now we begin to map what's going on in the community from just what the species changes are as we go out through. Here's the dry season over here. Here's the start of the rains right here. And here is the peak, the low after that, because these adults that came out here have all died off and their kids are growing here. The kids don't get caught by Malaise traps. And then when the kids produce the next generation of adults, you get this enormous number of individuals again. What they look like inside the forest, here's 3 traps in the deep forest. Notice that these are orders, the colors are different orders. Notice that they look almost identical from all 3 traps. The striking thing is that the proportions of these orders are the same essentially in all 3 traps. 3 replicas, if you like. However, the species composition of this one and this one and this one are very different. In other words, at the ordinal level, there doesn't seem to be any change. At the species level, there's a lot of change, but it has nothing to do with the drilling site. Now, if we look at all 9 traps over the year. There's one who's way off base. Very different. Dry season, start of the rains, low larval level, and then back up to adults again. This is the edge. This is the number of species caught on the edge in 1 year. This is the number of species caught by the other two edges in the same 1 year. Why are they so grossly different? The green one down here is the road edge. And since the engineers collaborated with us by making the road only one lane, what happened is after the road was put in, the forest grew back over the edges of that one lane so much that this trap was effectively in the deep forest. So it's basically the same as the ones that are very far away, 50 meters and 150 meters away from the road or the platform. The blue one over here is very interesting. This is trap number 4 on the margin originally. This is trap number 5. You can see how far apart they are. That's about what, 2 meters, something like that. This was in the deep forest. And these are the buckets of alcohol from those 2 traps on the same day. All right. Now, about 1.5 years into this project, the electric company decided they had to expand the platform. So in expanding the platform, they very kindly said, all right, we'll move the trap that's on the edge to the new edge. They did so. That brought it right up to this one right here. So suddenly there's an edge next to this trap. It's also next to this one. But notice something. The collection bottle for this trap is towards the forest. The collection bottle here is towards the open sunlight. The outcome is that this trap here suddenly became the same as that other trap that's on the very edge, who by accident had its bottle towards the platform. This one always from the beginning has had it away from the platform, and it's always been very low. i.e., where the traps are matter a great deal, and how they're oriented matters a great deal. This is the edge we're talking about. Here's the platform here, here's the original forest here, and who occupies this? Because this is very different taxonomically, which we can tell from the barcodes, than the deep forest is. That's the corn rootworm diabrotica. These are plants that grow in pastures. This is from the agricultural countryside, growing right here. This is that edge we're talking about. What else is on that edge? This butterfly normally lives high in the sun on the edge of the forest, the upper edge of the forest, the canopy. But he comes down to this level now here, just as though it were canopy, and lays an egg, which produces this little first instar caterpillar, which is under that piece of leaf, which then grows into a nice big fat caterpillar. The second point is that the first point is agricultural community here. Second, it's a community from up here down to there. And third, it's those drifts that come from the inside of the forest. So you get 3 different communities contributing to the community of insects on the edge. Here in this particular point, I want to acknowledge the chunk of money required to this barcoding, $25,000 and then more, came in great part from the Walder Foundation in Chicago, the Japanese government for development, Japan, the Canadian government itself, and 2 donors, longtime donors, who have both died and are gone. But they put together the big chunk of money needed to do the barcoding for all of this. We're not yet done, but almost done. Okay. Now, let's look at what Malaise traps are catching, okay? This blue circle right over here is 1,140 species caught by Malaise traps over 30, I'm sorry, reared from caterpillars. Reared from caterpillars over the last 38 rearing years. which involves about 900,000 caterpillars, something like that. This is the number of parasite flies, the species, sorry, number of parasitic fly species that have come out of those caterpillars. Good. So this is a big chunk of known fauna. Well, we don't have a name for them, but what we have is an interim name and a barcode and a bin. Okay. Now we look at the Malaise traps, run in the same forest. 68 Malaise trap years. They've caught 595 species. In other words, they've only caught half as many as we have reared from the same forest. All right. So when you go out there and put one Malaise trap down in some place and you think you're catching the fauna, you're only getting a fraction of the fauna. Okay. And of course, that's true for all the things that live in the litter, the things that live deep underground, okay. Or the things that are too smart to get caught in a Malaise trap, or the things that are too small to get in a Malaise trap. Now, what is the relationship of these two? The PL12 Malaise traps, those 9 traps, caught 30%, 38% of the known tachinid fauna. The known fauna is the combination of what's caught in the Malaise traps plus the reared. And then we're certain there's a lot more, because we certainly have not reared all the caterpillars here, and we certainly have not had Malaise traps in all the habitats long enough. Okay, but it's a start. But what it tells you is these only are getting a piece. So if you just put down Malaise traps and you know anything about this, you get a very, very different viewpoint. And of course, we can ask this for the edge. We can ask this for the deep forest. And now we can do that numerically with just sitting here in a chair. Okay. These are wasps, which have the same parasitic lifestyle as those flies do. But in this case, the female wasp generally puts the egg inside the caterpillar, whereas often the flies put it on the outside or even on the leaf next to it. But the simple point is here, you get that same result with parasitic wasps as you do with parasitic flies. And it turns out you get the same result with any group you work on. Okay. And I should add that this has a huge number, as needless to say, of undescribed species. We have, I'm just crudely estimating that there's at least 3,500 and probably a lot more species that are undescribed of the known things, which are 313, 3,300. And of these, so far, we've only been able to describe about 700 species in 17 years by standard taxonomic processes. And I should go back to this slide and point out that we've only been able to describe out of these 1,500 undescribed species, only 185 over 10 years. So the point is that if the real world wants to know what's going on in there, they can't wait 10, 20, 30 years before they decide, hey, we should pay attention to this. You have to have results now. So what are some of the results? Just another ecological observation on our part as tree huggers, if you like. This is the platform we're talking about. This is where the Malaise trap is, and there's another 50 here and 150 out there. But in fact, it looks terrible, but in fact, what it is a landslide. That's what a landslide does. It clears the forest away down to bare bedrock. And there are landslides everywhere in national parks all the time. And nobody complains at all, because we all know what this disturbance does. It allows more species to survive in that forest than if that disturbance had not occurred. There are other kinds of disturbance. This river right here is an analog to those landslides. Here's the bare piece in the center. Yes, it's occupied by fish and insects, but the point is it's very different from the surrounding. This is the edge, and this is the forest interior. This is right inside of the ACG, and we're delighted to have it as a conserved wildland. That drilling site we were just looking at is right over here on the base of this volcano. But in the society itself, this is extremely important. This is the National Electric Company, the arch enemies of conservation. on their own, deciding to work with the conservation area, which is over here. And this is their slide, not ours. I wouldn't even have thought to be bold enough to make this slide. They made that slide showing that they are now working with. And up here in the corner is the logo of the electric company and the logo of the government national parks, or the ACG itself. That's the first time there's ever been a slide in Costa Rica with those two logos on the same picture. This is the president signing off on a decree about BioAlfa to allow the barcodes to be public domain, circulated anywhere. That's what BOL requires of their information. And now Costa Rica has officially made it legal to do that. Okay, that means the whole world can use the barcodes that are in bold for their own usual taxonomic and other kinds of purposes. It's the only tropical country in the world that has done that. The other end of this sociological pyramid are the parataxonomists in the field, the people doing the actual work of gathering these specimens and organizing them and sorting them and getting them off to where they can go to BOLD or to go to a taxonomist or to both. Okay, these are people who mostly made it through 6th grade. There's one PhD in this photograph, a couple of master's degrees, and that's it. They manage the whole process there on the ground. This is one of those parataxonomists explaining what she's doing to 15 Colombians who've come to Costa Rica, to the ACG for a week to learn or see what this is happening and what it's doing. Gloria is explaining to PhDs, commercial people, high-level government people, high-level university people, what she's doing. She's only had a 6th grade education. This is happening. This photograph is 1 week before COVID hit Costa Rica. What she's showing them is this. These are species pages written and developed by the parataxonomists themselves by having to learn how to work on the web, produce the program, put it on the web, take it down, repair it, put it back up again, all by themselves. I learned the names of these species by reading about them in books. Today's generation in Latin America certainly is not learning about them by reading about them in books. Books are termite food for these people, for the world as a whole. They're learning on their web. That's where they're learning. So if we click now, this over here on the left is from the web. If we click on one of these things, we get the author. This is a fish killer, a professional fish killer from 3 decades ago, who has spent his life now becoming a parataxonomist. Now, with BioAlfa, we got permission from the government itself to teach park guards how to use Malaise traps. They go home to their parks with their Malaise traps and they set them up and they do this inventory. This is an image they put on the web. We had no idea it was even coming or what it was or anyone didn't see it beforehand. These are 2 park guards in the National Park, Tortuguero, who are installing their first Malaise traps. And they were very proud of being able to do this. So they put it up on the web for their special day of forest. What we see in this photograph is not the Malaise trap. What we see is a gun right there and another gun right there. These are armed, gold badge park guards, traditionally charged with being police. They are now being subverted into becoming parataxonomists at the same time. The outcome of that is that now there are Malaise trap samples from all over the country, sitting in a freezer in Guelph, waiting for the funding to be able to do the remainder of that first year of barcoding, which will allow the system itself then to be able to say, oh, the insect density here is the same as here, but the species are totally different. The species from here are totally different from here on the edge of the Atlantic versus the Caribbean, or are they all the same? So suddenly they have a real numerical way besides birds, trees, monkeys, lizards, snakes, to evaluate their own biodiversity. The red stars are private individuals or NGOs who joined in to collaborate with this, paying their own bills, okay? This is paid for by the government. But the government, after COVID, in no way has the money available to be able to barcode all these things, even at $1 a specimen. But we take a lesson from Canada. The Center for Biodiversity Genomics put Malaise traps in schoolyards all over Canada, like 150 schools for a year. And they caught more insect species of insects in 1 year in Canada than Canada had registered in 300 years of standard taxonomy and standard collecting. Well, Costa Rica has high schools all over the country. So why not have them do the same thing? And it's taken us several years to accumulate enough grants to be able to start with one first trial high school this January. Agriculture is a very big piece of Costa Rican life, and they have the same situation that the geothermal project had. They have heavy duty industrial impact coupled with adjacent forest. So now they are now paying the bill themselves to ask the question of how different is our agricultural landscape from the forest next door. So here you are here. This is on the edge of a pineapple field. Here it is. Here's the same trap over here. And this is the pineapple that you ate for dessert yesterday, produced by Dole Pineapple Company and Banana Company. And they are managing the trap and they are paying the bill for the barcoding. The whole point about this is to facilitate Costa Rica to bioalphabetize itself, to become bioliteracy for everybody. This slide was generated by a Costa Rican, not by me, all right? And here are the sort of takeaway messages from how Jenny viewed this thing. Know all the wild multicellular organisms that are in Costa Rica. Accomplish this by DNA barcoding, coupled with all the other information you can get. And put all this information on a public and available database, one that's available to everybody, every school child, every industrialist, every tree hugger. I don't care who you are, but that's like literacy saying all words in the vocabulary, all words in the dictionary are fair game. You can use those to develop those things for yourself in your own way, for your own purposes. And again, that's the concept behind BioAlfa. And again, this was chosen as a logo because it's known to everybody from bottom end of South America all the way up into Mexico. Thank you.